This is a really quick QuEST review for all of those people who filled out some of the common questions on the Retake form. So one thing that a lot of people wanted help with was labeling this diagram. I had to redraw it, and I'm not the best artist in the world, so I apologize in advance. But we're going to go over how to label this drawing. First thing that I want us to look at is the backbone of our DNA. There are two main parts. There's your deoxyribose sugar, and there's also your phosphate group. Let's see if phosphate group will actually pop up on my screen for me. So you have those two main parts. Your deoxyribose sugar is a 5-carbon sugar. That's why it's shaped more like a pentagon. Your phosphate group is shown as just a circle. And they're held together right here with a covalent bond. That covalent bond is really strong. It helps DNA hold on to its structure even during replication. The other thing that we want to look at is what's in the middle. So you have these rungs of the ladder like this, and they're made of base pairs. Each individual part of the base pair, each half, is a nitrogen base. And those nitrogen bases are able to pair up and to be held together by these hydrogen bonds. They're drawn for us as just these dashed lines in the middle because they're relatively weak. Helicase is able to break them apart, and that's really important to the structure of DNA. The other part that's important is that these three things, your deoxyribose sugar, your phosphate group, and your nitrogen base, they make up the monomers for DNA. So each time that those three are matched up with each other, they make a nucleotide. And nucleotides are our monomers for DNA. All right, going on to our next question. A lot of people asked about this table, and they were confused how we were able to fill it in. So let's take a look at that. We have to use Shawgraff's rule. So he told us what our base pair rules were, which is really important for us to remember that adenine and thymine pair together, and cytosine and guanine do as well. This table tells us the percentages of DNA for a sample that we're studying. So the whole thing, since these are percentages, should add up to 100%. So let's go through and look at these. It's telling us that there's 20% thymine. We know since thymine and adenine bind together, that must mean that there's also 20% of adenine. And since this is a percentage and all of these numbers need to add up to 100, we can do a little bit of math to figure out how much cytosine and guanine there is. So let's do that together. We're going to take our two 20% for adenine and thymine and add them up. That gives us 40% total for those base pairs. Then we can subtract that from 100 to get the percentage of the other two base pairs of cytosine and guanine. But we're not done yet. We need to divide that in half to get just the percentage of cytosine and just thymine. So that's how you get cytosine and thymine for these other two boxes. Take a minute and pause this video and you try to do number 2 on your own. You can check your answer by adding all the numbers up and making sure that they add to 100. All right, hopefully you figured this out. Pause the video and actually tried it out on your own. Let's go over it together. It tells us that there's 15% cytosine. That means there also has to be 15% for guanine. So we have 15% for both of those. Let's do our math. 15 plus 15 is 30. Take away that percentage from 100%, and you get 70% left over for our adenine and thymine. Now, if we divide 70 in half, we get 35% for the individual. So 35 for adenine, 35 for thymine. And if you add all those up, 35 plus 35 plus 15 plus 15 equals 100%. So we know we did it right. All right, if you still have any questions or if you're confused about anything, please let us know. We are happy to help you. Have a fantastic day.